Lactose operon. Introduction. In prokaryotic genes, the genes are mostly present in clusters and they are regulated by a single promoter. That is, when multiple gene expression are controlled by a single promoter and single terminator, it is called as polycistronic operon. It is mostly found in prokaryotic cells. This operon is polycistronic in nature due to the absence of the spacer or the non-coding sequences between two consecutive genes. Operon concept was first discovered in E. coli by Jacob and Monod in the year 1960. Structure of LAC gene. It consists of promoter for regulatory gene and structural gene. Next is the regulatory gene, that is I gene, also called as LAC repressor gene, encodes for repressor protein that represses the transcription of LAC operon. The operator, which is the negative regulatory site, it is called as negative regulatory site because of binding of the LAC repressor protein to it, which inhibits the activity of the RNA polymerase. Hence, there is no transcription and translation in the structural gene. Next is the cap site, which is called the positive regulated site, to which the catabolite activator protein binds and enhances the transcription and translation process. The structural gene consists of LAC-Z, LAC-Y, LAC-A genes. Regulation of LAC operon. The lactose operon of E. coli encodes for the enzyme beta-galactosidase, which hydrolyzes lactose into galactose and glucose. The LAC operon consists of three cistron or DNA fragments that encodes a functional protein. The protein encoded by the cistron may function alone or subunits of larger enzyme or structural proteins. The Z gene encodes for beta-galactosidase. The Y gene encodes for a permease enzyme which facilitates the transport of lactose into the bacterium, whereas the A gene encodes a thiogalactosidase transacetylase enzyme whose function is still unknown. All three of these genes are transcribed as a single polycistronic mRNA. Polycistronic RNA contain multiple genetic messages, each with its own translational initiation and termination signal. The activity of the promoter that controls the expression of the LAC operon is regulated by two different proteins. One of the protein prevents the RNA polymerase from transcribing, which is called the negative control. The other enhances the binding of the RNA polymerase to the promoter, which is called the positive control. The negative control of LAC operon mostly occur in presence or absence of the inducer, that is the allolactose. First is in absence of inducer, that is the allolactose. The protein that inhibits the transcription of LAC operon is a tetramer with four identical subunits called as LAC repressor. The LAC repressor is encoded by the gene, that is LAC-I gene, which is located upstream of the LAC operon and has its own promoter. Expression of the LAC-I gene is not regulated and very low level of the LAC repressor are continuously synthesized. Genes whose expression is not regulated and the expression is continuous, it is called as constitutive genes. Next is the presence of inducer, that is the allolactose. Mostly in absence of lactose, the LAC repressor block the expression of the LAC operon by binding to the operator. The operator consists of a specific nucleotide sequence that is recognized by the repressor, which binds very tightly physically, blocking the initiation of transcription. The LAC repressor has a high affinity for lactose. When a small amount of lactose is present, the LAC repressor binds it, causing dissociation from the operator, thus freeing the operon for gene expression. Substrate that causes repressors to dissociate from their operators are called as inducers, and the gene that are regulated by such repressors are called as inducible genes. The next is the positive control of lac operon. Although lactose can induce the expression of the lac operon, the level of expression is mostly very low. The reason for this is that the lac operon is subject to catalytic repression or the reduced expression of genes which are brought by the growth in the presence of glucose. That is, when glucose level increases, the catabolite repression occurs. Glucose is very easily metabolized, so is the preferred fuel source over lactose. Hence, it makes sense to prevent expression of lac operon when glucose is present. The strength of the promoter is determined by the ability to bind RNA polymerase and to form an open complex. The promoter for lac operon is weak and consequently the lac operon is poorly transcribed upon induction. There is a binding site upstream for the promoter for a protein called as the catabolite activator protein, that is the cap protein. When this cap protein binds, it distorts the DNA so that the RNA polymerase can bind more effectively. Thus, transcription of the lac operon is greatly enhanced. In order to bind, the cap must first bind to cyclic AMP, which is a secondary messenger synthesized from ATP by the enzyme adenylate cyclase. In presence of glucose, circulating cyclic AMP level are very low and consequently the initiation of transcription from the lac operon is very low. As the glucose level decreases, the concentration of cyclic AMP increases, activating the cap, which in turn binds to the cap site, stimulating transcription. The cap cyclic AMP complex is called the positive regulator. The E. coli mostly exhibit various abnormalities in lactose metabolism. Some of the mutants express the lac operon genes constitutively, whether or not lactose was present in the medium or not. Such mutants are called as constitutive mutants. So now discussing about the mutation of the lac genes and what are the phenotypic effect. First is the I gene. When I gene is mutated, it shows constitutive expression because the operator is never closed, whereas mutation in operator gene results in constitutive expression because the repressor protein cannot bind. If promoter is mutated, no expression of the operon occurs because RNA polymerase cannot bind. The LAC-Z gene mutation results in no glucose or galactose production from lactose, whereas mutation of LAC-Y gene results in no induction because lactose will not be taken into the cell. These are the references. Thank you.